Hello, this is Marcia, and my flow cytometry method is FISH, fluorescent in situ hybridization. Here are my contents. We'll go through the definition of FISH, the principle of FISH, the procedure of FISH, applications of FISH, and then how it applies to hematology. So the definition: FISH, or fluorescence in situ hybridization, is a laboratory technique used in order to identify or view a specific DNA sequence on a chromosome in cells or in tissues. DNA segments are recreated in the lab and are tagged with a fluorescent dye, and they're known as probes. These probes will be added to the patient sample to bind with the DNA. They will light up with the fluorescence when they are viewed under a specific microscope. This method is used to identify a gene location on a chromosome, count the number of genes, look for chromosomal abnormalities, and help diagnose diseases. FISH can also be used for other types of things, as we will explain later. This is a diagram of, a simple diagram of how FISH is done. And I will describe the steps in the next slide. So the principles of FISH. Step 1. The first step includes preparing the patient sample by fixing it to a slide. Step 2 is using heat. The DNA strands will denature, causing them to separate. So they're going to separate into 2 single strands of DNA instead of the double strands. The next step is to prepare a short sequence of single-stranded DNA to match the patient sample and then attach the fluorescent tag. This is called the probe. The next step, the probe will be added to the patient sample and incubated in a moist chamber to help with the hybridization process. This is the bonding of the single-stranded DNA to make double-stranded DNA, as is going to be shown in the next slide. The next slide will also have how it's denatured and then how the hybridization happens. After the hybridization, the unbound probes will be washed off and the slide will be dried. Finally, they're going to examine the slide under a fluorescent microscope to identify any abnormalities or gene expressions. These are the denature and hybridization process pictures. So in the first part on the top, we have the double-stranded DNA of the patient sample, and on top of that there's a DNA probe that they've created in the lab. With heat, the double-stranded DNA is going to separate or denature, and then during the hybridization process, the DNA probe will be placed into that segment of the gene. And with this, there is that fluorescent dye, the markers, that will then light up under the microscope for them to see. These are just some of the applications of FISH and how it's used. One of the first ones is the detection of chromosomal abnormalities. They can detect different chromosomal aberrations. So could be mutations, it could be deletions, any kind of abnormalities that might be happening in the DNA sequence. There are probes for specific genes, and they can locate fusions, abnormal numbers of chromosomes, and the loss of chromosome regions. FISH can also be used for gene mapping. Researchers can visualize the genetic material and can map it in individual cells. They're able to map specific genes or sections of genes. A third one is the identification and diagnosis of genetic diseases. Scientists are able to detect a variety of structural abnormalities in chromosomes, like small deletions. There may also be medium deletions or moderate deletions. And the Prader-Willi syndrome is an example of one of those moderate deletions. Some more applications of the FISH method. They can also include prenatal diagnosis of chromosomal aberrations. This is to determine the health or abnormalities in an unborn child or fetus. For example, there are some. 2 fluorescent signals are normally observed on regions 13, 18, and 21 of those chromosomes. 3 signals of fluorescence indicate an extra copy of the chromosome. And the chromosome 13 is Patau syndrome, chromosome 18 is Edwards syndrome, and chromosome 21 is Down syndrome. And these are just some of the ways FISH are used to determine any health abnormalities in the unborn child. It can also be used for the detection of cancer. It can be used to detect chromosomal abnormalities with cancer. The HER2 positive breast cancer has extra copies of the HER2 gene. This method is used to locate those extra copies. Chromosomal aberrations are also in 60% of AML and 65 to 90% of ALL. They are a more sensitive technique in diagnosing acute leukemia than G-banding. Here's an example of the FISH application in hematology. It can be used in the detection of cancer. FISH can be used to identify chromosomal abnormalities, including different types of cancers. Translocation of the chromosomes can occur when a part of the chromosome breaks off and reattaches to a different chromosome. The FISH method can detect abnormalities in the chromosomes. The acute myeloid leukemia has a unique subtype. It's called APL. An example is when chromosomes 15 and 17 translocate. This abnormality of chromosomes is acute promyeloid leukemia. APL accounts for 10 to 15% of AML cases each year. Here are my resources, and thank you, and I appreciate you guys for listening.